Thank you all for attending. Today we have three speakers for the webinar, and these speakers are Dr. Ben Coleman from JGI, Dr. Chris Tregel, and Muskan Kapoor from Iowa State University. And I'm really excited to introduce these speakers as I have been collaborating with this group for the last couple of years on gene expression biocuration efforts, especially on EBI Atlas. So one of the main goals of this webinar is to introduce the community to the upcoming new Ag Biodata Working Group on single-cell RNA-seq biocuration to focus on single-cell data and metadata management in plants and animals. So this new working group will be for a year, starting January next year. And specifically, the goal of the new working group is to have a compilation of minimum standards on sample metadata, cell types, and minimum considerations which are required to analyze and store single-cell RNA-seq results. So this webinar aims to introduce well-established methods and approaches to analyzing single-cell RNA-seq data, as well as the most common practices for data storage and need for better standards in the field and what that could potentially enable. So we have three speakers. So I will start with Dr. Ben Coleman. So Dr. Coleman is a computational biologist with a background in plant biology, phenotyping, plant microbial interactions, and single-cell RNA-seq. Dr. Coleman works as a project scientist at JGI, leading single-cell atlases on plants, especially on sorghum and switchgrass. Ben is also associated with the Plant Cell Atlas team. So next speaker will be Dr. Christopher Tregel. He is a distinguished professor in the Department of Animal Sciences at Iowa State University. His group is working in the area of porcine functional genomics and bioinformatics. Dr. Tregel is also an executive board member of AG2PI, Agriculture Genome to Phenome Initiative, and founding co-chair of the FANG consortium. So FANG, you might know, it is a function annotation of animal genomes, which has over 450 members in over 20 countries, with the goal of determining all functional components of the genome of all domesticated animal species. Muskan Kapoor is third speaker. She is a third-year graduate research student in Dr. Tregel's lab at Iowa State University. Muskan obtained her undergrad degree in bioinformatics from India. Her research is centered around the bioinformatics application of single-cell analysis with a particular emphasis on tool development and data analysis methodology. So now the floor is yours, and I would like to request everyone to please mute yourself. If you have any questions, please feel free to write in the chat box, and we will have a QA session at the end of all the talks. Thank you.
Thanks. So I'll kick things off here. Cool. Thanks, Sunita, for the nice introduction. My name is Benjamin Cole. I'm a research scientist at the Joint Genome Institute at Lawrence Berkeley Lab. And today, what I wanted to do is kind of give an overview of single-cell technology and also discuss some considerations that have to be made for storage and reporting and analysis of the data and availability of metadata. So to start, I kind of wanted to take a step back and ask, why it's important to why it's important to consider cells and cell types and single cells in plants in particular, which is my area of background, although I think this is universally interesting to all fields of biology. So what makes plants kind of interesting is that one of the many things that make plants interesting is that they're kind of fixed in place, but they have to kind of react to their environment. And so this environmental stress can come from a variety of different sources, from physical and biotic, and they have to just kind of deal with that in situ. And you might imagine that plants aren't just monolithic entities. They're composed, like every other organism, multicellular organism, of a variety of different cell types. And each one of these cell types can react to their environment in different ways. You might imagine, the vascular system might respond differently to a heat shock than the epidermis or the photosynthetic organs of the plant. So I think it's important to consider how all of the different components of the plant are are interacting and responding to the environment and among themselves, and also how they develop. So, I like to think about these key questions for uh, our group is, how many cell types are there in plants? They've been described anatomically, but are there more that we aren't really understanding? How do they develop? How do they respond to the environment? And how are they specified? Um, and so a couple of years ago, a really revolutionary technology kind of entered the molecular biology field, and this is single cell sequencing. And I won't belabor this point because I feel like a lot of you are probably very familiar with this, but you can basically take a tissue and decompose it into its component cells. You then use a microfluidics device to tag and then create libraries from those cells so that you can decompose the complete transcriptomes of thousands to tens of thousands of individual cells from a population in a single assay. This data matrix, a gene by cell matrix, is then processed to produce these maps, and they don't necessarily have to be resolved in simple 2 dimensions, but the goal is to resolve them into maps that we can then use to understand how cell types are specified and kind of what they're doing, and hopefully begin to infer trajectories, either developmental or reaction trajectories of those cells from that experiment or multiple experiments. And so since the advent of single-cell RNA-seq and other modalities, the plant community has kind of lagged behind the animal, especially the mammalian community. The number of plant single-cell RNA-seq data sets has really started to take off, and I think is really at an inflection point of kind of exploding. So a recent survey that I did along with members of the Plant Cell Atlas, and I kind of want to shout out to the Data Management and Infrastructure Committee at the Plant Cell Atlas, who kind of helped collate a lot of this information. There are over 50 studies. I think now it's up to like maybe 75 or maybe even 100 studies or so that have generated single cell data. And this was organized around 300 samples across multiple platforms, and including a lot of different species, not just the favorite models like Arabidopsis or maize. And this also encompassed a bunch of tissues and treatments. And it's really started to take off. The first plant studies were published in 2019. There were just a handful, around 5 or so. And now it's really started to explode. And so I think it's important now, before the field kind of takes off and starts generating tons and tons of data, to really consider what data is being reported and how to kind of describe the data in a metadata scheme. And so kind of saying, I just want to say, the multiple data sets are now being kind of integrated together into these sort of meta data resources. And so I think, considering data and metadata is also really important for stitching data sets together so that we can use the power of the community of all these people who are doing all of this really, really hard and not to mention expensive work to generate this data. We want to make it ultimately useful for everybody. And so I presented this last week at a Plant Cell Atlas workshop, and I think that it's an interesting, motivating scenario to think about, that I like to think about when generating my own data, is you sort of have executed a single cell study. It's cost the lab tens of thousands of dollars, multiple years of work and analysis and optimization and all that, and you kind of just arrived at the end. You've hit submit, you sent this off to nature, you finally figured out what auxin does in plants, which is like this really big nebulous question. And you've produced this massive data set, and to celebrate, you take this backpacking trip to Iceland, but your laptop falls into a volcano, and all of the data that you have there is completely lost. And so the question is, you come back and reviews are good. They just want you to tidy up a few plots and maybe produce a few more. But how prepared are you to recreate your own analysis? And I think that's like, the baseline level of how much data and metadata do you have in a resource that you can then go back to and access and then recreate all the things that you did. And so there are other reasons why it's important to have accurate and comprehensive metadata descriptions. One of them is it's useful to test out new pipelines and tools. As I mentioned, these large-scale meta-analyses are enabled by the availability of metadata. It's useful to migrate workflows from one platform to another. Maybe you're, moving your local analysis onto a high-performance computing environment, and you need to kind of transfer all of your workflows over there. And it also helps to improve reproducibility. So I wanted to kind of take a step back and say, what is actually the data and the metadata that are important for single-cell analysis? And so when I say data, what I really mean is the primary information that's generated by these assays. This usually includes FASTQ files that can be pretty large. I think the recommended sequencing depth for a single-cell RNA-seq provided by 10x Genomics, which is one of the more popular platforms out there, is about 200 million reads per 10,000 cells. So that can be a pretty large file. And also information directly processed from these FASTQ files. And what I mean here is like this data matrix of a gene by cell, describing all of the transcripts, the abundance of all of the transcript in all of the different cells. What I mean by metadata is pretty much everything else. So this is information describing the data, and this can include experimental considerations like how the sample was generated, how in plants, how the plants were grown, how they were treated, any quality control metrics that were applied to particle isolation or treatment prior to entering into the single cell workflow, any insight that you gain from the analysis. So a lot of groups spend a lot of time annotating their single cell data, to contain information like cell type or pseudotime trajectory metrics. And it's really important to have that able, to be able to report that information, be able to link it back to the primary data source. This is usually structured with a handful of metadata terms that should be fixed. I think that there's a lot of great efforts out there to start to fix these into a more standard ontology, but I think some more work needs to be done there. And ideally, all of the data and metadata should be machine readable at minimum and human readable if possible. So I kind of wanted to go through in a little bit more detail like what data and metadata are generated from like a typical single cell analysis workflow. So these range from the sample collection, these, as I was saying, describing the growth conditions, experimental factors, relationship to other samples if you have a more complicated experimental design, describing replication strategies, et cetera. Particle type that you're looking at. So single cell can be done on either cells, whole cells, or more increasingly nuclei, which also contain a complement of transcripts that are more nascent. And the choice of particle has implications on the type of analyses that you can do downstream. Sample QC, anything that was done to say that those particles were high enough quality to proceed with library prep. And then, metadata associated with library preparation, importantly, like the kit that you're using to generate single cell data, the version index, all those things that you would need to take that primary data set and transform it into a more processed data set. Information about the sequencing. So this is, this is where the big data comes in, like the actual FASTQs. Those need to be available somewhere that can be accessed. And then all the metadata associated with sequencing, including the platform, how many counts, read quality, accession numbers, and which repository the data is provided in. And providing all this information makes it easier for automated tools to kind of go and grab that data and work with it. And then finally, as I was mentioning, it's really important for anything that come out of the analysis, any metadata that comes out of the analysis to also be reported. And this is, for primary analysis, just transforming those FASTQs into gene matrices. These are typically encoded as either just a sparse matrix. An H5 file is becoming more popular now, or alignment file types like BAM or SAM. I think at minimum, like this barcode matrix should be documented somewhere. And then analysis metadata. So this is like cell type, pseudotime, other metrics that say maybe whether a cell is high quality or low quality. Those types of things should also be provided, ideally as like a CSV file. And then, also all the code that was used to generate all of the analyses and the analysis environment itself can also be very important. So you can run the same exact analysis pipeline on two different computers and get two different answers often. So, I mean, most of those differences are trivial. But if you're trying to recreate a plot, for example, using a lot of these more fancy methods for dimension reduction, they're very reliant on stochastic methods. And so, just doing it a second time, can produce like a wildly different visual layout, which can be problematic. So having your environment kind of documented as like a Docker, for example, can be extremely helpful. And so, kind of an overall theme, like it's helpful to have all of this stuff in an organized way, and also to have identifiers that can link all of these data types all the way through the entire workflow. And you can never have enough documentation. And again, where possible, make things at least machine readable, available via fixed URL to make aggregators able to access your stuff, and avoid locking data behind paywalls at journals or elsewhere. So I think some unique challenges in plants. I guess a challenge overall is there are a variety of different repositories with different metadata and data formats. So this can make it challenging for meta-analyses, for example. In the plant community, there's also a lot of different versions of the same plant genome. So they could use the same assembly, but either the gene models are slightly different, or the annotations, the gene names or identifiers for those gene models can be different. And that also makes it really challenging to compare, two different analyses that are done on the same data set, mapped to the same genome, but using like a different annotation, for example. So, documenting like which annotation version you're using, or ideally like just including like that entire annotation file with your analysis can be helpful. Also, there are some metadata terms that describe the experiment that I find are oftentimes missing, like the time of day for harvest, which is in plants, and I guess animals too, can have a really, really big influence on the overall transcriptome. And also, growth conditions and growth substrate are also important to report, which I think affect the plant community in particular. So I'll kind of breeze through the rest of this because I think I'm running up on my half hour here. But so some of my favorite tools to kind of handle all of these different considerations include really excellent resources at the NCBI and GEO. I know that there are similarly great resources at the EBI, including the ENA and the single cell expression atlas. A lot of the workflows are done in R, so I like to have organize my code into this R package called targets, which kind of tracks analyses as you develop them. It's similar to SnakeMake for those of you that are more familiar with Python workflows. And it encourages functional programming, which can be really good for documentation and reproducibility. GitHub is a great resource for housing your code because it's can be open to the broader community, and it's easy to kind of grab code snippets from GitHub. It also makes issue tracking and all of that a lot easier. And then Docker Hub, as I mentioned earlier, is a really nice tool to sort of codify your analysis environment so others can reproduce your stuff. So, in the plant community, a lot of different visualization browsers have cropped up over the years, and a lot of these are really, really amazing. So shameless plug for the JGI, we have one of the Arabidopsis resource at Phytosome. Uwe Ohler's group at MDC Berlin and Philip Bethany's group at Duke have this AVREX browser that hosts their data sets in Arabidopsis as well. There's a cool browser at TER that uses the bar resource to describe single cell, at least for this this Arabidopsis root data set. That's nice to kind of be available to the whole community. And then again, single cell expression atlas at EBI, and now this single cell plant DB at Nanjing University is attempting to aggregate a lot of different data sets together. And there are many others of visualization browsers. So there can be issues with each and every one of these things, but they are really, really useful tools. And I think efforts to standardize and bring them all kind of together to work harmoniously could be really important. And then on the metadata side, there are kind of existing and emerging tools that are growing more adept at handling metadata. One is this MageTab format for single cell that the EBI is kind of spearheading also through Annotator. And then a new one for describing a sequencing library from Leo Pachter's group called seqspec. Is kind of like a standard metadata format that is used to describe any sequencing assay, and single cell included. So that could be useful for reanalyzing data without having to go fish for all of those different parameters that are specific to each library. And then so at the Plant Cell Atlas, we've been thinking about a lot of this stuff and are very much interested in working with AgBioData working group to kind of bring a lot of these ideas to the community, so that the PCA is a network. If for those of you not familiar, network of hundreds of members of the plant community, and we are organized into subcommittees focused on specific aspects of the PCA, and our kind of data management infrastructure group, together with Noah Falgren, are working to identify needed standards, specifically with data and metadata that are specific to the plant community. And here's something that I think working with AgBioData could be really useful. And then we're also kind of going out and collecting some useful metadata for just kind of aggregating everything together as another community resource that we can provide. So with that, I'll close out and thank all the folks at JGI involved in generating and analyzing and visualizing a lot of the single cell stuff. And also special thanks to the Plant Cell Atlas community, Suri, who is our kind of is heading up the PCA, as well as Noah, who co-chairs this data management infrastructure committee with me, Nick. Probart for also playing a leadership role in the PCA, especially Selena Gomez, who Selena Gomez, no, that's not right. Selena Rice, who is kind of keeping all of our heads together, as well as everybody else on the PCA data management infrastructure community. And so I'll close this out and hand it off to Chris or Muskan, unless there are questions.
Yeah, so currently there's no questions in chat box, so we can, maybe Chris or Muskan, you can go ahead.
Okay, great. Let me just share my screen. So is it working? Looks like it.
Yes.
So what we thought we would do today is talk about. I should say thanks very much for the introduction earlier. Appreciate that, and appreciate the opportunity to discuss our small seed grant from AG2PI. What we thought we would do is, what I'm just going to do is spend just a couple of minutes describing the initiation of this seed grant, and then Muskan will describe basically go through quite a bit of the nuts and bolts of how she's been working with EBI and developing some new pipelines. The title is Single Cell Genomics Data Incorporation into Agricultural Genome to Phenome Research by Building a Fair Data Ecosystem. And so just to give you the talk outline, we'll cover 2 points: improvement of pipelines in order to ingest single cell data into publicly available databases for sharing and further analysis. And second, development of a visualization tool or platform for lab level research projects, and talk about 2 of those, one using JBrowse and another one using Shiny in order to visualize the data in 2 different ways. So as an introduction, we really want to think about, and Ben has indicated that, I'd say plants are behind humans, but animals are, at least farm animals are behind plants. So this was a summary with an idea toward plants and animals, but especially animals. The users currently are on the left here. Basically, all the steps are really manually performed either by the user or by the curator. And the user then needs some level of computational training to create the data and metadata, transferring it to the web portal, and creating or using the pipelines. And the whole idea, the long-term goal is to have new biological insights. But the problem is that this process is not fair. The best practices and for information and for training are really quite scarce. And there's no centralized knowledge portal or annotation portal, especially for livestock species. And these resources are poor, as I've said. On the other hand, the single cell genomics infrastructure is well advanced in biomedicine, as has been said already. I would point out the Human Cell Atlas data coordination platform as a good example of the well-developed resources in biomedicine. And that's, of course, part of the Human Cell Atlas project, which is a very large project, extremely well funded. And the HCA DCP is integrated with Terra, which is also a large community and data resource. It's a cloud-native workbench for computational biology, not just in single cell, but lots of genetic data, and basically it's the data warehouse for the Broad Institute. And it was developed by Broad and Verily and Microsoft, and it houses tools for single cell genomics analysis. And so what the idea is, can we leverage these resources to benefit the agricultural community? And we proposed this and got funding for Muskan to do some work in this area using a pilot scale project level analysis, and try to test ingestion of the agricultural single cell RNA-seq data that we have, and convert it with the processes that Muskan will talk about to transfer it into the HCA DCP using their standards. And then this is a diagram of sort of what we would call our future vision. The idea is that we had 2 aims in our project. One is to demonstrate via a small number of data sets, can we bring the data in and can we ingest it into the HCA DCP, which then will allow the data to be automatically transferred to the Terra computing environment. And then that would allow us to use the Terra workbenches so that the users could then use a much more automated approach to create biological insights. And then in addition, sort of in parallel, we wanted to develop some interactive tools that will allow especially the agricultural livestock community to use these data sets with tools that are sort of more community specific. And I guess what I wanted to do now is transition to Muskan, and she's going to tell you about all the processes that she went through. So I need to stop sharing. And then she can pick up.
Can you see my screen?
Yep, looks good.
Okay, so next we are going to focus on the problem of agricultural public data, which lacks sufficient metadata for the efficient reuse. So this table represents a single-cell metadata, which is a crucial way to describe a biological sample from which genomic sequences are derived, and it should always align to the FAIR principles. In the single-cell RNA-seq, the general workflow unfolds through 3 different portals, which are plant cell atlas, animal cell atlas, and the public data. In the plant cell atlas part, the user submits their data to ArrayExpress or ArrayExpress via annotator, and the raw sequencing and processed files are submitted to the ENA portal. In the public databases, there are mainly 3 portals used, which are ArrayExpress, ENA, and GEO. Within the animal cell atlas, the user submits the data either to the FANG portal or one of these portals. From the FANG portal, the raw files and sequencing files are submitted to the ENA by the user itself. The user performs validation checks and summation in the FANG portal. So next, in the plant data path, manual curation and validation checks are performed by the curators so that the data is transferred to the single-cell expression atlas. All these steps are manual and are time-consuming. Similar is the case with the public datasets, where the manual curation is performed to transfer the data again to the scEA. Whereas in the animal cell atlas, we tried to make this path more automated by transferring the animal data, which is in FANG, to the HCA DCP ingestion service. One point of doing this was to compare the species, the 2 species, which is animal and human, which will be important for further analysis. So we created this automated path where the data from the FANG goes to the HCA DCP ingestion service through ingestion validation of the JSON files. To facilitate user access and analysis, we transported the data. So the single-cell expression atlas data is linked to a Galaxy platform to visualize the data further and analyze in the Galaxy portal itself. Galaxy portal offers a range of single-cell tools developed within Galaxy. And from the human cell atlas, the data goes through Terra using an automated process transferred by the data wrangler, which we can say like in curl command. The data goes to Terra, and within the Terra, there are a list of tools, interactive app, and batch workflows, which allow user to analyze, visualize the data, and also create new tools or develop new standardized workflows. So this figure represents all the general workflow, where you can see the animal path is much more automated, which was the main objective in our project. And this triangle represents the manual metadata effort. For example, the users have to submit well-defined metadata into the FANG portal and have to go through rigorous validation checks. And then the metadata manual effort reduces as we go down the lane. So it's more quicker, and the data can be shown within Terra in a much faster way. Whereas in the plant path, the amount of metadata submitted is very less. For example, user submits the data to ArrayExpress or GEO or through annotator. It has to go through rigorous validation checks and manual curation by the curators, and they have to go back to the paper, look at some metadata, or contact the authors of the paper to go down the lane. So this is much more slower process. Next, we are going to focus on the animal metadata path. This is an overview where we can see the single-cell RNA-seq data and metadata is added to the FANG portal. So in order to make FANG more useful to the single cell, we added new rule sets of the single cell data within the FANG portal. Then we created the JSON files for those single cell, validated those, ingested those, and transferred it to the HCA DCP ingestion service through an automated path. From the HCA DCP ingestion service, the data goes to Terra, which is a computing environment, and new metadata tables were created. And either the data wrangler or the users can see their data in the Terra. So from Terra, you can have the list of tools and analysis app or standardized workflows to further analyze your data. So this is in-depth discussion of the previous steps discussed. First is users or the experimental biologists submit the data to the FANG data portal. So in the FANG data portal, we updated the rule sets according to the single cell rules, which were namely sample metadata, experiment, and analysis metadata. This is a pictorial representation of the FANG data portal. As you can see, there are 3 sample, experiment, and analysis spreadsheets, and the single cell rules are listed below. You can always choose only mandatory fields and fill in the metadata table or download and see what example templates look like. And you can perform validation within the FANG portal and submit it to the ENA and the FANG portal. Next. Once these files were updated, the next step was to validate the JSON files. So we created these JSON files according to the updated rule sets, validated them, and this is what a JSON file looked like. So JSON file stands for JavaScript Object Notation File. And this is an example of the analysis file where you can see this is the name and the attribute listed for analysis file and the value filled in by the users. So once these files were validated and ingested, next step is to perform the ingestion to the HCA DCP ingestion service. So this ingestion is performed by a data wrangler. And this step, when we were trying to perform the ingestion, we faced few errors which were based on the schema difference between the HCA DCP and the FANG data portal. So the schema difference first was there was no described by field as per the HCA rules and no float value were taken. Second, there was no connection of analysis file with other 2 files. And third, we needed a full project information tab. This is like separately, but we needed a full project indicating all those files are connected together. And fourth, only mandatory values were considered, whereas in FANG, as you would know, there is an option for optional and recommended values. So how we solve these errors? First, we had described by fields pointing towards the JSON metadata rules of the FANG portal. Second, we used already published ENA files through an FTP command to establish a connection between all those files. Third, we used each sample as a of FANG to align with the HCA schema, thus creating a full project information tab. And fourth, we changed the schema according to the HCA metadata schema so that the only mandatory values are considered. As you can see, this is an example of FANG HCA JSON schema transformation. And the first, this is the described by field pointing. This is a part of project information tab and analysis file connecting all those files from the ENA, and only mandatory values are considered in this part. So once the ingestion was performed, again, the step was to do validation on those ingested files. So again, this was done by a data wrangler, and we validated the files and showed in the HCA DCP ingestion service. So, but with this, we had to do some modification with the JSON files. For example, we created a GitHub page where you can see we added scripts so that we can clean, delete, or add further JSON files if there are any. Second, we created a page where you can see a new summation for another FANG HCA dataset. Third, we created valid graphs which specified how the data is interlinked to one another. And fourth, we had these metadata tables created for further use in Terra, which is shown in the HCA DCP, the project, essay, and the file metadata. You can see the file is validated. I'll show this figure more clearly in the next slide. So this is a project information tab where it is done by the HCA DCP ingestion service. This was a failure because the metadata was invalid. And when we ingested and validated these files according to the HCA schema, we can see the files are submitted and it was a success, and all the processes, the sequence files are loaded along with the data. Next is, once the data is shown in the HCA DCP ingestion service, the next step was to transfer the data to the Terra computing environment. In the Terra, you can have various analysis portals, storage and method packages, and visualization and data sharing platforms. So once the data is transferred to the Terra environment, There are a list of method packages, for example, Magic, Fate, Slingshot, Cumulus, along with the analysis portals, for example, Cell into Gene, Cytoscape, and many more, which are single cell portal, Omni browser, Cell browser, where the user can use these method packages and portals for further analysis. That's what completed our project on the animal side. On the plant side, we have single cell RNA-seq data and metadata, which is transferred to Array Express, and the raw sequencing files or the processed files are transferred to the ENA portal, or they are already available in Array Express or ENA portal. As Ben mentioned, there's another portal called Annotator, which allows for data submission that is linked to the Array Express. So from Array Express, manual curation and validation checks are performed by the curator, so the data is transferred to the single cell expression atlas. And again, the curators have to go back to authors or check the rules so that the metadata can be aligned with the single cell expression atlas rules. From this, an API retrieval is done so that the Galaxy analysis tools and resources can be used by the single cell expression atlas data. This is a high-level overview of how the ingestion is performed within single cell expression atlas. So from GEO, manual curation is done, which is then transferred to the single cell expression atlas. And from that, a retrieval accession is done of the data to transfer it to a computing environment called Galaxy. So in this, you can copy paste the accession number from single cell expression atlas here. You can choose what type of matrix you want and use all the available single cell tools within Galaxy for further analysis. This is again the in-depth discussion where the main goal is to transfer this data to single cell expression atlas. So it starts with the user who submits soft format in Array Express, and it would always be an add-on if the user submits marker gene and cell type info. Then the curators have to convert the MAGE tab, which is an IDF and STRF file, validate the converted MAGE tab, and then generate config scripts so that the MAGE tab is configured and moved to the single cell expression atlas. From the single cell expression atlas, experiment accession retrieval is performed, which then transfers the data to a computing environment called Galaxy. So we tested and developed prototype tools to visualize the output of single cell data from Terra on a genome browser and compare it across tissue and cell populations. So this is a JBrowse view showing tracks of the PBMC single cell RNA-seq data. So the single cell RNA-seq data shows a bar chart for each gene, depicting gene expression level for each cell type. And these are the bulk samples for this particular data set. And when you right-click on the PBMC single cell RNA-seq track, you can see the category score in the cell type column, which shows the gene expression level, which is called score for each cell type. You can also see the name, length, and position of that particular gene. Moving forward to the visualization tool for exploring single cell data, which is called a Shiny Piggy, a web-based Shiny application, which runs on any modern browser and has an R framework. Also, it is hosted on Microsoft Azure platform. This allows single cell level transcriptomic study of pig immune tissues in the PBMCs and will be an important resource for improved annotation of porcine immune genes and cell types. So the tool is hosted online at this link, and you can always scan the barcode and visualize the outputs from the tool. So this is a small video. As you can see, the first is the metadata for this particular tissue, which shows the 4 tissues, number of cells post-QC, number of features, and number of clusters. And then along with other metadata, for example, the bone marrow gene list, which shows the cluster of every cluster of bone marrow tissue and genes listed with that. You can also see the spleen, thymus, and the lymph node gene list and the link to the paper. So the second page is the genes page, where you can see the gene expression of this particular gene across all 4 tissues. For example, these are the 4 tissues: bone marrow, lymph nodes, spleen, and thymus. As you can see, the expression of the CD3 is the maximum in the thymus according to the scale of 1 to 4. After this, there comes a gene expression among all the clusters in all these 4 tissues. So this is a volume plot showing the expression level across all 4 tissues. And there's a plot showing the color specified for each cluster. And then there's an integrated SORAT object for all those tissues, showing the expression of a particular gene, which is CD3E, in all these 4 tissues according to the scale. You can always see another gene, for example, PAX5, and see its gene expression for tissues, clusters, and the integrated SORAT object. So for the conclusions, we created an automated data ingestion pipeline to move FANG data to the HCA DCP and on to the Terra environment. So the JSON files were created and updated for FANG and HCA portal. Metadata tables were created for a new creation, which was for FANG HCA data portal. And a GitHub page was created for training of data wranglers so that they can know how the data was created, how we perform all the ingestion pipeline. And next, we participated in the curation and ingestion of Arabidopsis single-cell RNA-seq data within the single-cell expression atlas. And we developed a prototype tool to visualize single-cell RNA-seq data on a genome browser, which is called JBrowse. And lastly, we created a Shiny-based tool called ShinyPiggy for lab-level single-cell RNA-seq data visualization. The future scope would be we further intend to build these tools to construct a scientist-friendly data resource and analytical ecosystem and facilitate single-cell genomic analysis through data ingestion, storage, retrieval, and comparative annotation across agriculture species. For the animal data, we'll test to use the data ingested in the HCA DCP further in the Terra environment. For plant path, we'll try to reduce the manual curation and validation needed to transfer the data. And for the JBrowse, we'll make it publicly available for the users to perform interactive visualization. And lastly, the ShinyPiggy will add the cluster annotation functionality to the tool. Now, the future vision would be single-cell cloud-based analysis tools. For example, there is a Terra workspace for exploring the single-cell data and inbuilt pipelines for standardizing the workflows. The user can use the existing workflow, or he or she can create the new workflow or the pipeline within the Terra workspace. Next is Galaxy. So it retrieves the data through single-cell expression atlas and use the inbuilt tool for streamlined analysis of single-cell data. One major difference between these two is Galaxy is free for the public use, whereas Terra has some initial cost, which is very low for analysis and storage of the data. With this, I would like to thank all the collaborators and the funding agencies, and we are open to any questions if you have.
Thank you, Muskan. Thanks to all speakers. It was wonderful. And there's one question from Nick in chat box. So you have done a wonderful job, Muskan, and you have gotten the FANG data into Terra. And it is really important. You have also built Shiny app on the top of that. Will this ingestion process be performed for all FANG data going forward?
Yeah, so we created another, like a GitHub page where new data wranglers can learn and train themselves to transfer another data set from the FANG portal directly to the HCA DCP ingestion service.
Mm-hmm. And hopefully we can also do the same thing for plant data, wrangle into single cell expression atlas or Terra. So then it will become easier both for researchers and curators. So yeah, so it's wonderful.
I think the big problem, though, Sunita, is that the big difference, as Muskan pointed out, the big difference is FANG puts all of the metadata creation primarily on the user or the submitter of the data, and And that's partly because it's a FANG community. It's not all of the public. Whereas I think the plant community has to decide whether they're going to follow that. If they don't, the pipeline will be very small, right? I mean, it's primarily just the manual curators within the single cell expression atlas at EBI that has to do it then.
Yeah, absolutely. I agree with you. And this is something that we have to, have discussion during new working group, single cell biocuration working group. So this is also one of the objectives that we will be discussing on it, because this is really important. And we do have FANG model. So we can see that how much we can do it, how much we can copy this. Yeah. Any other question? Is there any other question from the audience? I do see some comments, but there are no questions, unless if I have missed any. Yep. So Leonard has mentioned that it is a general comment that it seems like still a lot of data cleaning required. Yes, absolutely. Doreen has any, yes, Doreen.
Hi, guys. I was just curious, how portable are your Shiny apps to be able to use on other sites for the stuff that you just showed for the single cell?
So right now the Shiny app only works for one dataset, but our future would be like the users can add their data and visualize for the gene expression and the cluster functionality. But yes, you can visualize the data through the website. It takes around 20 seconds to load, so that might be slow.
Thank you, Muskan. So I'm curious for Chris and Muskan and the people who are working on the agricultural animals and that discussion you were having about making decisions. Do you have any sort of summaries of lessons learned and what you would make as suggestions to the community? Same thing for Ben, on as we're thinking of moving forward.
Discussions about what, Doreen?
So in the case of the way that the animals are handling it, and the choices, for instance, to push much of that on the submitter, but actually giving them tools to do that. Do you have thoughts on how successful? So I think it would be useful to share with this community how much scaling you've been able to do with that versus perhaps some of the ways that things have been handling. So with the existing tools that are being used with FANG right now, how many samples are being submitted and what's like the timeline for it? Because it kind of gives us an idea, or I'm hoping that you can communicate to this group, if we can shift the responsibility onto the submitter, how this would support scaling and why this might be a better path as long as they're given tools to support that activity, right? So, because I think that's part of the discussion here to bring that up. And since we have two diverse groups right now, and even though I know that there's going to be a single cell group, I think discussing the success or perhaps failures with that would be a really valuable sort of like, elevator speech. Give me like the 3 best things about that and the 3 worst things.
Well, I can start, and Muskan has submitted some data via this method, so she can chime in too. Basically the structure is a set of Excel spreadsheets is provided to the submitter, and they fill them all out. And then once they've submitted their data and they have the accession numbers, those get put into the spreadsheet as well. And then that spreadsheet is used as a tool to automatically connect the metadata and the data together in the data portal. I would say
and Chris, on that end there, is there a validator of that spreadsheet? So do you have to have previously, very constrained, controlled vocabularies? And is there a path forward to update those? Has that been successful? Have there been some bottlenecks with that? Like, I'm trying to get a feel of how many QC tools do you have to support what goes in?
It fails if it's not correctly filled out. So, I mean, there are validators. I'd have to ask Muskan is it a defined vocabulary, or is that, I think part of the problem is sometimes there's misspellings. So what I think would be better, which we don't have, and I've talked with EBI about that, and it's a large lift for them apparently. But what I would like to see is drop-down menus within the Excel spreadsheet. So you have to choose 1 or 10 things as opposed to type something in, and that would have to be maintained. That's their main problem, is that they could create that once, but then it would have to be altered every time. It was a pretty big lift for us to add additional fields for single cell data, because it started with bulk data. Muskin, it's typing, right? So you can make typing errors basically.
So firstly, the farm data portal has validation checks where you can fill in the data and then perform validation. It will automatically tell you if that's right or not. And secondly, there are it's a spreadsheet, and you can see the rules or the control vocabulary in the farm data portal itself. The downside is user have to go back and again to fill the spreadsheet and look at the rule sets that are defined by the farm portal. Whereas a plus point in the annotated platform, which is developed by the single cell expression atlas, is the same as Chris mentioned. They have drop-down menus, and you can see what all vocabularies they require.
And you also know what is expected. It's not always completely clear what is being requested in the FANG spreadsheet. So
For EBI,
if we could come up with one where we could use it for both plant and animal and not have to revisit that, it would be great.
Definitely, yeah. Because for EBI Atlas, we still need to have some validation later on as well. Once we have done curation, and then later on, even if it's through annotator, later on we have to do validation and configuration, and that is manual, and that takes a lot of time.
So it sounds like I'm just bringing this up as we're talking as a community together. So it sounds like there has been quite a bit of success with being able to scale in the animal community, people submitting that metadata, and that the templates are working. But there could be some opportunities to support development of some path forward to add new terms at an interim, and maybe provide some more software support to be able to have the autofills and things put in. And maybe you could even see, I don't know how long this RCN will exist, or whether this would be part of like a Plantium or a plant cell atlas like activities, but you could have committees or things that could be chairs that review that on a quarterly basis and update, or something, terms that come in that might be very sustainable.
I was just checking, and it looks like there's over around 3,000 data sets in the FANG data portal right now, but most of those would be bulk RNA-seq or chip-seq type of data sets.
But Chris, those are still impressive numbers.
Yeah, and the portal, I mean, the spreadsheet works well. I mean, it's I think the problem, the issue is the community has to decide that the user will be the one to make sure the data is well described, and that one of the big problems that the FANG community or leadership of the FANG community was always concerned about is there's a fine line between if you require more and more things that the user has to do, then you may not get them to submit it to the FANG data portal. They'll maybe just dump it in the easier path. You want to have as much of the data in there as possible. So you don't want to require too much. You don't want to have too many rules. But if you don't have the rules and a good approach, that's that's sustainable, then you don't have much of a system. So it's a difficult question. How much do we want our members to have to do before we lose them?
Yeah, Damien mentioned in the chat that there are a couple of automated tools for kind of transforming metadata spreadsheets, and I guess there was a webinar on that I missed that. But that could be another way, instead of having a million different items in a dropdown, to getting like the user to submit a first pass, having it go through a like a transformer that will hopefully report back to the user exactly what is their original term and what it got changed to. And that would take a little bit of the, because I mean, I think, well, if you're submitting like 5 samples, it's not a big deal to go through like a couple of drop downs. But if you have like a similar schema for describing cell level metadata, where you've got thousands of cells and you have to do like a drop down for each one, and that for example, for a cell type, that drop down can include dozens of individual terms. That could be pretty cumbersome. And so, streamlining the experience for the user and making it as absolutely easy for them as possible, I think is critical. Otherwise, it's just not going to get adopted. And another thing that might also encourage users to provide a lot of metadata is also maybe add a benefit to them for doing so. So, by submitting through this portal, like maybe they can get access to like this shiny visualization browser, for example. So they don't have to develop one on their own. And, they could just feed all of their data into that and, report that in whatever paper they're preparing.
It's a good point.
That might be a way to encourage people to go through the extra steps to get their data on. Because I think like, establishing the tools is, it's challenging, and there's a lot of stuff to think about, but I think even more challenging is enforcement and adoption across the community.
Yeah, I think one of the major carrots was that their data would be included in meta-analyses of community manuscripts and projects. That was one of the major carrots, but that's a long-term carrot often, right? It may be a year or 2 or 3 years before that comes to fruition. So it's a good point.
Yeah, thank you everyone. So it was wonderful talk by all of you, and we really had very good comments too. And now we are out of time. So, but yeah, so if you guys, anyone who has not joined this new working group, RNA-seq Biocuration working group, and if you are really interested, please feel free to contact Anarita and send your email and more information so that you can be part of this working group. Thank you.
Thank you. Bye-bye.
Bye-bye.